So anyway, let's talk about the material, the incubation. So a few more eyes. So why do we incubate? The short answer is that incubation provides us controlled environment. Which means the results can be reproducible. They are either reproducible from one experiment to another experiment, or they are reproducible between the labs. Okay, so I do something, and Anishka does something in the same conditions, and our results basically match. Make sense? No, not really? Make sense? I mean, blink once if yes. Isolation. So there are three methods for isolation. Pour plate, spread plate, and streak plate. So let's deal with pour plate and spread plate. In this case, if you have some kind of liquid medium with microorganisms growing in it, you have several tubes that you can make dilutions in. Does that make sense? So say this is your original tube. So you make 10x dilution, and then you make 10x dilution of a second tube, and you just produce serial dilutions of the original sample. Does that make sense? So you progressively reduce the concentration of your microbes in the sample, and then you can either, if this liquid is agar, you can pour it in the petri dish. It will solidify, and you will see colonies. This liquid is the broth, is the medium. You can pour it on the agar, spread it around with a hockey stick like this, and observe growth. Obviously, the more diluted your sample is, the more separated the colonies are going to be. Does that make sense? We're not going to do either spread plate or pour plate because it's a bit of a labor-intensive and time-consuming. But we will be doing a streak plate, which is the classic method of isolating microorganisms. Why we do it? Imagine you have a yellow colony on your plate and you're really interested in it. But you have to be sure that this yellow colony is actually an individual organism and not the mix of white and orange colonies. Does that make sense? So what you do, you pick that colony with a sterilized wire loop, okay? And then you pick a clean agar plate, and using that wire loop, inoculate a small fragment of that agar plate. Then, you're not sampling anymore, anything. You just sterilize your wire loop over the flames. Now look, you pick a little bit from that first streak and spread it across the clean section. And then you sterilize the loop again, pick a little bit here and spread it across the clean section. And then sterilize a loop again and spread it across the clean section. So with each streak, you essentially dilute your sample on the surface of that dye. Does that make sense? So with each streak, your colonies become more and more separated. So in the end, you will be able to see if that yellow colony actually represents a single species or there are multiple species. And once you've done a streak plate and you have these individual colonies, you operate on two main presumptions. First, that each colony originates from a single cell, which I can tell you up front is wrong, not necessarily. you may need more than one cell to produce the colony, okay? And all cells in the colony, you remember colonies from your handbook, all cells in the colony are the same, which is also not exactly right, because due to the mutations, there can be slight differences. But for our intent, for instance, we often use, when we say, we have what we don't say usually we have 1,000 cells in the suspension. We say 1,000 colony-forming units. Okay? Not cells, colony-forming units. We're good so far? But these presumptions, they don't have to be entirely correct. But you say, for our intents and purposes, those presumptions work. Are we clear? Okay. After isolation, we do inspection. What does that mean? Size, color, shape of the colony. So you can say it's a round, or it's filamentous, or rhizoid, convex elevation, okay, or it's undulate, or the edges can be rough, the edges can be sharp, blah, 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 all that kind of description. Blue, red, whatever. And also put here microscopy. So you collect microorganisms from the colony, you stain them, and you can say gram-positive, gram-negative, acid-fast, flagellar stain is positive, so on and so forth. Why we say that this is an inspection? Often, at this stage, you can say, with a pretty great deal of confidence, what microorganism you're dealing with. But in most cases, you can't. So you need to perform the fifth step, which is identification. And in terms of identification, we'll be talking about three main types of identification methods, which my goal was to pick methods that relatively well comprise the spectrum of what you can run into the clinical settings. By no means this list is comprehensive, but it's pretty good in terms of, what kind of result you can get from the lab, so you understand how this result was obtained, what are the benefits and the limitations of each method. We good so far? So these will be immunological methods. I call them genetic methods and biochemical methods. Okay? So we're going to talk for about 40 minutes, I think, maybe less than that. We're going to talk about these methods of identification of microorganisms. The first set of methods will be immunological. And before we start, I want to remind, I hope you kind of have a maybe vague remembrance of what it all looks like. Okay, get some colors because it would be good to have colors. Okay, I want to remind everybody of such a concept as antibody. Okay, so imagine that we have a mouse. Okay, and we inoculate this mouse with some kind of antigen. Antigen means that this molecule, whatever we stick in the mouse, is foreign to the animal. It's not from mouse. That makes sense. So since it's not from mouse, mouse produces what? It produces antibiotics. Okay, so it will be a molecule which will be able to specifically bind an antigen. Does that make sense? So this is antibody. This is antigen. We're good so far. So all immunological methods are based entirely on the recognition of antigen by antibodies. In those immunological methods, you can detect, in some cases, you want to detect an antigen. In other cases, you want to detect an antibody. Does that make sense? Okay, good? All right. So, our first method that we're going to talk about is, it's called Western blot. So let's start with a sample. Imagine that this, right here, is the sample of, let's say, patient's bronchoalveolar lavage. We're talking about coronavirus patient. So the wash from the patient's lungs. And we want to know the patient's lungs contain antigen. And put it big question. Okay. So what we do, this bronchoalveolar lavage contains a whole bunch of different proteins, okay? One among those proteins can be an antigen, an S protein. From coronavirus, okay? So how can we detect it? Well, first and foremost, if we will just try to add antibodies that will bind this S protein, it's not going to really help us because it's all mixed, you know, and the concentration of S protein can be so low that even if antibodies bind, they will not be able to produce a signal. We won't be able to visualize. So first, we need to separate this S protein somehow from others. We can do it by separating S protein by mass. To do this, we prepare what is called a gel, and we'll load our sample on the gel. And then we apply electrical current so that proteins start to move from negatively charged to the positively charged side of the gel. Now, which proteins will go faster? Smaller or bigger? You have a forest, you have a bunch of kids, really scrawny kids and really fat kids. Which kids will make it faster through the forest? Scrawny ones, right? Same goes with proteins, okay? Smaller proteins will move faster, and bigger proteins will stay behind. Make sense? So you separate proteins by mass. And let's say your presumably, if, and I'm going to put it in a dash line, if there is S protein of coronavirus, it will be here. Remember, you're just separating the proteins. You can see nothing. Seriously, nothing. When you run the gel, all you see is the colored markers of the molecular weight of the proteins. Your sample, clear. You can say, well, we have antibodies. How about we'll just throw antibodies in there and see when they light up. The problem is that the gel is very fragile. Does that make sense? It's like working with a gel. So you want to transfer your separated proteins into something more workable, more sturdy. So this is when the word blob comes in play. In this case, you take a gel, that's my left hand, and you take a membrane, just like a piece of paper, you put one on another, and again you apply electrical current. And all those proteins from the gel get transferred on the membrane. You can picture it something like this. So you've got a gel, okay, and you've got a membrane, and all those proteins, they get transferred like this. Electrical current. So what you end up with, instead of a gel, look. Now, you have a membrane with all the same proteins, right? All the same pattern. And presumably, somewhere here, you have your S protein. Got it? But this time, you can work with that piece of paper. And what you do, you add antibodies right here, to that piece of paper. What these antibodies do? They bind to the target. And the target will be the S protein, right? So you can kind of picture the antibodies binding to the S protein. Does that make sense? Now look at this. Look at this quote-unquote piece of paper. Look at this membrane. Will antibodies bind to this band? No, because they are not targeted towards this band or this band. No, they will bind only to this specific band. Does that make sense? I'm going to leave the technicalities behind. You can detect this complex. You can make it fluoresce. It can produce luminescence. It can change the color, for God's sakes. The point is, you can see that band. Am I clear? And mind you, if there's no S protein, there is no band. Does that make sense? How do you know that this is your S protein? Well, first of all, you can have a positive control. Second, you know the mass of the protein from the previous study. So for instance, if this protein has a mass of 200,000 molecular mass unit, atomic mass units, then you have a special scale on the membrane. You can see where there is a 200, 100, 50,000, whatever. So you know where your band is positioned. And if it matches the size, bingo, you got it. Okay? So you can detect antigen using Western blot in virtually any kind of sample. Advantages. Okay? Let's put advantages on the board. I'm going to put advantages in blue to make it pretty. Western blot is sensitive. Okay? I'm going to put simple, although take it with a grain of salt. Okay? I wouldn't say I wouldn't say cheap, I would say not expensive. So it's not like $100 per sample. If we don't count human effort. Okay, so I'm not counting the price of, a person. Disadvantages. Time and labor consuming. My personal record. I was running 60 samples at one time. I did 60 samples in one day. You go nuts around it because managing all that stuff, knowing where is what, it's really, really hard. So processivity is what? Semi-quantitative. And I'm being generous here. Realistically speaking, we're talking more or less. Not even like 2-3 times more. It's very, it's not quantitative much. And we're going to make a really long acronym. Okay? Follow my lead. You find only what you are looking for. So the acronym will be, I'm going to make an acronym out of it. Okay? What does that mean? If the patient has something different, some other disease, but you're not looking for it, patient is still sick, but you still have no answer. Does that make sense? Okay? You understand that idea? You find only what you're looking for. If maybe it's not coronavirus, maybe it's flu, or I don't know, pneumococcal infection, or some other fungal pneumonia. Okay? That make sense? So can we fix some of this stuff? Mostly this and this. The answer is we kind of can. And the method will be ELISA, which stands for enzyme-linked immunosorbent assay. You don't need to know this particular breakdown, okay? So how does that work? Instead of having a gel, you have a plastic plate. Plastic plate about the size of a palm. And on that plate, on the picture here, I show you 3 individual wells. Does that make sense? Can you see this? 3 individual wells. At the bottom of the well, you have antibodies. Okay? So what you do, you add your sample of bronchoalveolar lavage or spit from coronavirus patient. Now look at this. Sample number 1 on the left has a lot of antigen. Sample number 2 in the middle is a little bit of antigen. And sample number 3 has none. Does that make sense to you? Good? Now, next question is, well, after we add antigen, we wash all the crap away. Now we need to detect it, to detect antigen. How we do it? We add another antibody that will also recognize this antigen, and look what happens. Okay, so if 2 antibodies here, the detection antibody. Okay. 1 antibody here, and no antibodies here because they have nothing to recognize. Does that make sense? Is that understood? Okay. Turns out each of these detection antibodies, they have this little attachment at their end that produces a signal. You see that light green thing? It produces a signal. It can be a fluorescence, it can be a luminescence, it can be a change of color. Now look at this. Which well, 1 or 2, will have stronger signal? 1, because it has more antigen, more detection antibody. Does that make sense? This one will have no signal whatsoever. So using this method, you can actually not just detect whether or not a patient has antigen in the sputum, but how much antigen the patient has. Am I making sense? So, sensitive, simple. I don't want to sound like arrogant, but like Western blot is somewhat of an art. Okay, I mean, it's a craft. ELISA, I can teach a 4-year-old who can use arms to do ELISA. If a person can follow instructions, that's it. So, sensitive, simple, not well, depends. Usually not terribly expensive, especially when we talk about mass-produced. Relatively fast, high processivity, okay, can be automated. There are machines that will do ELISA. Like, all you need to do is to put the reagents and the samples, then you press the button and you walk on. Makes sense. When I say processivity, we're talking hundreds of samples a day, if not more than that. Again, you find only what you're looking for. Does that make sense so far? We're good? Moving on? Any questions? Now, the last bit, I'm going to squeeze it in here, it's not something. Immunofluorescence. Okay? Now, the concept is very simple. So imagine you have a tissue sample with a few cells right here and there, okay? And there is an antigen in those cells. Okay, so you add antibodies. They bind to an antigen in the cells. And these antibodies, often they already have that little fluorescent bead at their end. Okay? So they will start to fluoresce. Does that make sense to you? Now, I'm going to shorten the advantages and disadvantages. Advantage, tissue sample. You can see a localization. Does that make sense? So you can look at the sample of a patient's lung and say infection occurs in interstitial cells, or in the type 1 alveolar cells, or in macrophages, whatever. Does that make sense? You can look at colocalization, saying, not just in the cell, but the virus is localized in endoplasmic reticulum or Golgi, whatever. Downside, again, you find only what you're looking for, and obviously it's not a quantitative method. We're good? Questions about immunology? Okay, I will kind of sum up what I expect you to know about these methods at the end. We're going to talk about the biochemical method now. And you may I hope that you watched the video about it. It's called matrix-assisted laser desorption ionization. Okay. Let's start with biochemical, which is, brace yourself, matrix-assisted laser desorption ionization, time of flight, mass spectrometry. It's a mouthful of an acronym. Often folks get terrified about it, but let me assure you, there's nothing to be terrified about, because I'm going to break it down to you, and you will see that it's really a fairly simple method. Well, not technical, because like the whole machine is just, it's insanely expensive, but conceptually, it's not that hard. So, matrix assisted. You have a special compound that forms a matrix, and you mix your matrix with your sample of interest. Now, we're going to get away from the virus, okay, and we're going to talk about some kind of bacteria, okay? So you mix your matrix with the bacteria. We're good so far? That's your matrix assisted. Nothing more. Pretty simple. Then you take laser and blast your sample mixed with matrix with the laser. Laser is really powerful here, like Star Wars powerful almost. So what it does, it blows apart this mix of matrix and bacteria. Are you following so far? Imagine that laser, like like in the Star Wars, laser just blows stuff in pieces. And these pieces include fragments of your sample. So these are fragments. Small, larger, large, small again, small, large. So laser desorption. Am I clear? And now the last letter, ionization. Turns out that these pieces have different charge. Okay? You see, they charge. Does that make sense to you? Does it or does it not? I just see like, Oh. Okay, so again, you have a ton of fragments, and each fragment can be characterized by two parameters, mass and charge. Does that make sense? And then these fragments, they fly into these devices, accelerating, so say electromagnetic field. Okay, so these fragments right here are going to fly through electromagnetic field. Does that make sense? Because they are charged, they're going to fly. Now let me ask you this. Which ones are going to fly faster? Small or large? Small. Second, remember, it's electromagnetic, so this question may be a little harder. Which ones are going to fly faster? With a small charge, with a big charge? Big charge. Does that make sense? So, now think of it this way. Think actually quite opposite. Think of the ones that are slower and therefore have longer time of flight. Does that make sense? You understand? The slow ones will have longer time of flight. So we can say that time of flight will be proportional to mass. Does that make sense? The bigger the particle is, the longer it flies. Are we clear? Got it? And it will be inversely proportional to a charge. Because the bigger the charge is, look at this formula. The bigger the charge is, the shorter the time of flight will be, the faster those particles will be. Does that make sense? So what you end up with, different particles will hit the detector at different times. Quite clear, isn't it? Got it? So in the end, what you can see after the detector is the graph where on y-axis it's intensity, and on x-axis it's mass to charge ratio. Okay? And you're going to see something like this. Each peak here corresponds to a specific particle. Or, a set of particles with a specific M to Z. Does that make sense? So, ionization. I'm like, okay, matrix assisted, the sample mixed with matrix, laser, desorption ionization, blow them up, giving them a charge. Look, time of flight, mass spectrometry. You have a spectrum, okay? So in the end, you get kind of a signature. You see on the graph? It's a signature. Turns out that if you do the same sample in the same conditions over and over again, then the signature will be the same. Does that make sense? So it's kind of unconventional, but basically imagine that, well, you have a cup. You drop it on the floor, and it splits in 50 pieces. So imagine that if you drop the same cup in the same conditions, exactly the same way, pieces are going to be identical all the time. Does that make sense? So what you do next, you take Bacillus anthracis, okay? You run it through the machine, you get a signature. E. coli, Salmonella, all microbes that you can lay your hands on. And you create a database of the spectra, the signatures. That makes sense of that. You have a patient with unidentified microbial disease. You grow the infectious agent in the pure culture. You load it into a machine, and you get some kind of a signature. And then you run this. You run the signature against your entire database. And if you have a match, it'll pop up. Does that make sense? It's basically like fingerprints. So this method doesn't have the disadvantage of you find only what you're looking for. Because if your microbe is in the database, you got it. Does it make sense to you? You see what I'm saying? Disadvantage? Well, few. I'm not going to put it on the wall. You need pure culture. You can't just take a blood or a sputum. People are working on, getting around it, okay? And the machine costs 750 grand. So, okay, now, compared to a Lysa machine, which you probably can get on eBay for like a couple of grand. We're good so far? Any questions? Okay, so we're going to get to genetic methods. And then we're going to review our results from last week, and then we're going to take it back. Okay. So genetic methods, there will be two. One is going to be, and I'm going to draw only one, which is going to be PCR. It's called polymerase chain reaction. That's a famous PCR. And that's how we do most of the coronavirus testing when you have that brain scoop. Okay. So this is how it works. If you want me to kind of plug in coronavirus, so say people do that brain biopsy on you, then put the swab in a special collection buffer. Coronavirus has RNA gene. So first step that you will have to do, and it's all done in the same analysis, you want to convert RNA into DNA. Does that make sense? You can do it. It's not a subject of our conversation right now. Let's say you've done it, you have DNA now, okay? So you have a DNA copy of coronavirus genome. Are we clear about that? So this is our DNA copy of coronavirus genome. Double-stranded DNA, okay? So now, I can tell you this. If you will search, you can get a sequence. We do know the entire sequence of that coronavirus DNA. So we can find there is a software that does it. Like 20 years ago, people did it by hand, which was a bit dense. But you can find a couple of specific fragments in this genome that, are you familiar with the word flan? Being on the sides, just easier for me to use because it's a scientific term here. So two fragments that flan some kind of a part of the genome. So there's a fragment here and a fragment here, okay? So you see between them is a specific part of the coronavirus gene. Now, the question that you may ask is, but if we pick this fragment right here in the coronavirus genome, is there a chance that the same fragment exists in a human genome or mouse? Starting from a certain length at about 25 nucleotides, the chance is zero, literally zero. Like there's not enough DNA in the world. That makes sense? Like it is unique, guaranteed unique. Interestingly enough, it is. Okay, we're good? And on top of that, it's a combination. Okay, so what you do next? Remember that DNA has that feature, complementarity. A, T, G, C. A pairs with T and G pairs with C. Okay? So what you do, you synthesize two complementary fragments that are called primers. Now, that's going to be your primer 1, P1. That's going to be your primer 2. Okay? That makes sense? Next, you heat up your DNA. 95 degrees, so these strands, they separate. Because all the hydrogen bonds that hold two DNA molecules, two DNA strands together, they're all broken down. Does that make sense? Then, I'm going to say heat, and then you cool them down. But this time, look at this. Look what we have. This time, they separated already. So these fragments, the primers, P1 and P2, it's called anneal, they attach to the DNA molecule because of those hydrogen bonds. So you heat them up, everything spreads apart. Does that make sense? You cool it down, and primers, it's by chance to a little bit, primers will anneal to your DNA molecules. In the mixture, you have an enzyme that's called DNA polymerase. And what it does, it starts to extend a primer, making a copy. Does that make sense? So this is polymerase reaction part. Now, how many DNA molecules you have here? Like double-stranded DNA molecules in this step. How many molecules you have here? Double-stranded molecules. Huh? Double-stranded. It's one strand, that's another strand, so how many double-stranded molecules you have here? This is a double-stranded molecule. How many is that? One. How many double-stranded molecules are here? Two. So after one round of amplification, you double the number of molecules. Does that make sense? If I will do another round, how many molecules I'm going to have? From two, I'm going to have 4. If on those 4, I do another round of amplification, how many molecules am I going to have? 8. With each round, I double the number of DNA molecules. Does that make sense? In 30 rounds, I will increase the number of molecules by 1 billion. Like, not by, but 1 billion, more than billion times. Does that make sense? So, this method, first of all, and then you can detect that massively amplified specific DNA fragment. That's what PCR detects. Polymerase chain reaction detects and amplifies a specific fragment in the microorganism's DNA. Now, I can detect this fragment in a various way. It can be quantitative. So let's put advantages. Quantitative, right? Stupendously sensitive. And I'm going to say a few words about it. Inexpensive. If we do not count the price of labor, I once calculated for the lab that I worked in, how much it costs for a sample. I get something like less than $2 around one sample. Like, materials, okay? Can be automated. Disadvantage, behold, you only find what you're looking for. Right now we have PCR systems where you can look for, 16 different pathogens at the same time, multiplex PCR. But it doesn't, still, only the stuff that among those 15. Am I making sense here? So, few words about the PCR for coronavirus. You see this word sensitive? So when you go and they do PCR and they call you and say you positive, what does that mean? It means that after 35 amplification cycles, they have a signal. 35 means that they amplify the original number of molecules in your sputum or your brain sample. It's not brain, just so you know, by 500 billion times. Oh, not 500, about 30 billion times. That's insane, right? That's really insane. That means that in your sample, you had 5 RNA molecules. This is nothing, and that's the problem. These tests are so sensitive that they will pick up not necessarily the virus that makes you sick or makes you infectious. It just means that there's some virus in your, respiratory tract. You may be totally fine. You may not be sick. You may not be able to transmit the virus to other people, right? So we don't need that level of sensitivity, it turns out. Does that make sense? And you would say, well, why not? Why not? Because it takes time. Turnaround time. Any of you ever got tested? How much time did it take for them to come back to you? My son took 8. It's useless. In 8 days, we brought them to school and we traveled back. Like he was in school for a day already. So and now the idea that, and I urge you, if you want to, I can give you letters, we can talk and I can explain it more. But write to your legal representatives in the state Senate, in the US Congress, in the US Senate, to promote there are tests that are crappy and not sensitive in that way. That can be done in 15 minutes. And they can be done for $1 per person. And you need no equipment. All you need to do, familiar with pregnancy tests. Same idea. Only you're not peeing, you're spitting on it. So you spit on it, you wait for 10-15 minutes, and it gives you plus or minus. And if it's plus, you're infectious. If it's minus, maybe you have a little bit of a virus, but you cannot transmit it. Does that make sense? And that actually what can bring us to normal life. Because think of it this way. Before coming here, like maybe one of you right now is at the stage where you're just about to become infectious. Those crappy tests would sense it. And if you can do it in the morning, just, I'm positive. You call your boss and say, dude, sorry, I tested positive. I'm going to self-quarantine. And you keep testing. And once you become negative, you're good. You can come back to work. You don't need to sit at home. Does that make sense? So that is something that can be done and needs to be done, but unfortunately. FDA is a bit slow. Okay, another method. I'm just going to put the name. Sequencing. Take a sample. Okay. And you determine sequences of all DNA and all RNA in your sample. Everything. That make sense now? Now, advantage, it's not this. You literally find everything that's there. And that's the problem. Because if you know everything that's there, then how do you pinpoint what it actually is? You see what I'm saying? It's really hard to pin down the culprit. Sequencing has its own benefits. Even in the coronavirus era, recently I heard reports about the guy that was infected twice. Does that make sense? So how do they know that it was truly twice? because they had his virus, his sample, clinical sample from April and then from August. And they did a full sequencing and they identified that virus in April and virus in August, they were slightly different, like by a couple nucleotides in the gene. Does that make sense? But those are different viruses, so the guy, well, no, it's the same coronavirus with slight change. So they were able to say that repeated infection. By the way, second infection, no symptoms whatsoever. If the guy wouldn't get tested, he would never know. Nothing. Okay? We're good so far? Any questions? I'm going to pause the recording. So we're going to start our second unit, which is, I'm not trying to scare anyone, just, be prepared. This is the hardest. Second is the hardest. First is the easiest. Sixth is the second easiest. Fifth is third. Third, so I would put them 1, 6, 5, 3, 4, 2. Okay, so this one is the hardest unit because stuff that we talk about is fairly abstract. You understand what I'm saying? It's fairly abstract, and it requires a lot of imagination to understand what is going on here. But it is really important for our other conversations along the way for our discussion of different microorganisms and so on. So we're going to start with a discussion of basic structures of DNA and RNA. And we're going to talk about DNA first. So, features of the molecule. Double-stranded, anti-parallel, helix. So I hope double-stranded and the helix do not cause any trouble, right? Anti-parallel, we're going to figure it out as we go. So what is the elementary building block? The elementary building block is a nucleotide. A nucleotide, on a basic level, consists of the sugar okay, the nitrogenous base, we're going to put just B, and the phosphate residue, okay? So this part right here, the blue and red, is called a nucleoside, while this whole thing, is called a nucleotide. So nucleoside is just a sugar and the base, okay? The nucleotide is sugar and the base and the phosphate. Am I clear? Good. Now, first of all, why it's called DNA, deoxyribonucleic acid. Do you see this carbon here? There's no oxygen in it. This is why it's deoxy. That's important. There's no oxygen. Lack of oxygen makes DNA molecules very stable. Now, how is DNA molecule organized? So, turns out that sugars and phosphates are connected to each other. Okay? So this is another nucleotide. Does that make sense? And here, we can plug yet another nucleotide. I find it really important to draw it this way. Because 1, 2, 3. Okay? Does this organization make sense? Now look at this. First of all, all these carbons, here those are carbons, numbered like this. Okay? So 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. So basically what you have is that 3 prime carbon of one sugar is connected to 5 prime carbon of another sugar by a phosphate group. Does that make sense? Do you see that? Again, 3 prime carbon of one sugar is connected to 5 prime carbon of another sugar. Do you see that? By a phosphate, do you see that? 3 prime and 5 prime. They are connected by a phosphate. Got it? 3 prime, 5 prime, connected by a phosphate. So essentially, DNA molecule has a directionality. Are we good so far? Why it is important? When you read from left to right. When various enzymes read the genetic information in the DNA, they read 3 prime to 5 prime. Am I clear? Does that make sense? Now you have to appreciate that 3 prime, the sugars and phosphates, that's the backbone. That's the structure. What enzymes actually read, genetic information, is essentially in the bases. Okay? Because you see, all sugars are the same, all phosphates are the same, but bases are different. So there are 4 bases, A, G, C, and T. Okay? And they pair like this. Okay? Now, T and C are pyrimidines, and G and A are purines. You can see that pyrimidines always pair with purines and vice versa. Okay? Does that make sense? So the basis for pairs, and if we want to, so 2 questions. First, what holds the pairs together? Hydrogen bonds. There are 2 hydrogen bonds between A and T, and there are 3 hydrogen bonds between G and C. So GC is always stronger. Does that make sense to you? We're good. Okay. Now, if you think about a double helix, and I don't have, you see this decrepit model there in the corner with the tape right there, like really falling apart by that box. I don't know even if I can carry it around. This semi-destroyed. I don't know who handled it this way, but it's terrible. Need to restore it. Okay. Yeah, it's really in a bad shape, as you can see. So, bases are inside of the spiral. So in order to read them, you have to separate the strands. This is called a denaturation. You heat them up, you break hydrogen bonds, everything is exposed and can be read. Does that make sense to you? Now, the fact that it's double-stranded means that information that's in one strand is also in another. Hence the question, why the hell do we need 2 strands? One is a backup. If one strand is messed up, you can restore the information using the second. Make sense? We're good? Now, finally, what does that mean that the DNA is entire parallel? You have 2 strands like this. If one strand on top goes this way, 3 to 5, then strand on the bottom will go that way. 3 to 5. So they are anti-parallel like this. Always. Does that make sense? This will have a lot of important implications on the processes of transcription and replication in the cell. So far we're good? Okay. Denaturation makes sense for reading? Okay. That also has important implications. RNA. Now, what is the difference? First of all, the set of nucleotides, A, G, C, and U. So in the pyrimidine realm, instead of T, you have U. Second, the structure of the nucleotide is slightly different. So this is a single nucleotide. Now look at this. You see the difference between 2' position DNA versus RNA. You see the difference? This is why it's called ribonucleic acid. There is oxygen, hydroxyl group here. See that? That exact hydroxyl group makes RNA unstable, relatively speaking. And as you may remember from a cookbook analogy, it's fine because this is your photograph of a recipe. This is your scribble on a paper, okay? It's fine if it's gone in a few minutes, okay? Good? You see the difference? Generally speaking, and I want to say generally speaking, single-stranded molecule that does have secondary structure. Now, what is the secondary structure of an RNA? Let me demonstrate it to you. Imagine that you have 2 fragments of a single RNA molecule that are complementary to each other. 2 inverted repeats. So these blue parts are complementary. So what's going to happen with them? They're going to fall in the stem structure. Okay? There's going to be a loop at the bottom. This is called a stem loop, or sometimes called hairpin loop. These hairpin loops play an important regulatory role in the gene expression. Any questions so far? You good? Okay. Let's talk about different types of RNA, where DNA is in the cell, and start transcription conversation. So RNA in the cell, types. It's all about RNA so far. Messenger, or mRNA, carries out message. It carries the message from the DNA to the ribosomes for translation. Ribosomal RNA, it's the structural and functional element of a ribosome. Carries out some enzymatic functions in the ribosome. Transport, tRNA, it transports amino acids to the ribosomes for the protein synthesis. And microRNA, which is quite interesting because it regulates gene expression and controls the degradation of messenger RNA. So look at this. Imagine that you have a messenger RNA that for some reason needs to be destroyed in a relatively quick manner. Do you follow? So what would you do? There is a system in the cell, Foltz got Nobel Prize for that discovery, that produces very short, about 20 nucleotides, double-stranded RNA, which is kind of unusual. Now, this microRNA will form a complex with target mRNA. Can you see that? This complex right here. And microRNA will serve as a tag for degradation. So special system of proteins, if I remember correctly, they called Argonaut, but I wouldn't take my words to the bank. Special system of proteins recognizes this complex and destroys target mRNA. Does that make sense? Which gives you extra control over gene expression. You good? MicroRNAs are, people try to use them in the therapy of genetic diseases. I am not aware, which shows my ignorance, I am not aware of any actual diseases for which microRNA therapy has been approved so far. Maybe I should, search that. So far, it's okay. It's okay. All right, we're good? Any questions? So I told you before, I do not ask things beyond the stuff that's in the lectures, study guides, and my reviews. They don't touch each and everything. Some things are just minor. You watch the lectures, you cover those, okay? But study guides are absolutely crucial because all my questions and all my exams are written with a study guide open on another screen. I literally, it's not a figure of speech. I literally have a study guide open on another screen. So, what about DNA? We're kind of jumping back. In which form can DNA exist in the cell? We're going to get this done and swiftly move to transcription. So in the cell, DNA, well, first of all, let's figure out difference between bacteria and eukaryotes. I'm kind of avoiding archaea, so eukaryotes versus bacteria. So in eukaryotes, DNA can exist in the chromosomes and it can be extra chromosomal. So chromosomes in eukaryotes, usually more than one, which can be 4 or 72, like in the mouse, but more than one, and they linear. Extrachromosomal DNA in eukaryotes exists in mitochondria and or chloroplasts. Okay, we good? Does that make sense to you? Okay, in bacteria, chromosome, deliberately in the singular form. Usually one. Usually. There are some very rare exceptions. And it's circular. Extrachromosomal DNA exists in the form of a plasmid, which is a circular DNA molecule. plasmid is circular, which is smaller and usually confers some beneficial features. Does that make sense? Now, there's a video game called BioShock, and there are plasmids in BioShock. Never played it, but read about it. So basically, same idea. Plasmids give bacteria some extra capacity. Antibiotic resistance, ability to digest certain carbohydrates. Flagellum, ability to produce toxins. Does that make sense? If you take plasmid away, it does not kill bacteria. They can still reproduce, although they lose some of the features. Am I clear? We good? Still. Okay. Couple more things. Chromosomal DNA in both eukaryotes and bacteria is wrapped around histones, and that wrapping regulates gene expression. In lecture, I explain, like, it's not going to stop on it too much because it's a pretty straightforward concept. If DNA is wrapped around histone, genes cannot be expressed. If it's unwrapped, it can be expressed. Strictly speaking, histones are in eukaryotes, and histone-like proteins are in bacteria. So, okay, let's talk about transcription. Okay, so it's going to be, so let's start here. So you have double-stranded DNA, okay? This is the first nucleotide of a gene, and before the gene, there is a sequence that is called promoter. Very much like in the book, when you open a chapter and say a chapter says mechanism of transcription in prokaryotes, you know what chapter is going to talk about, but you don't know what the mechanism is. Do you understand? Like, you look at Avengers Infinity War, you kind of understand who the characters are going to be, but you don't know what the plot is. Okay? Does that make sense? So promoter just tells an enzyme that is called, that is RNA polymerase. Okay? It tells RNA polymerase, bind here. Okay? So RNA polymerase binds to the promoter. Now, here's what happens next. RNA polymerase, after binding to the promoter, slides to, well, about number 1. Let's put number 1 here. And unzips a DNA strand. Does that make sense? Because remember, you need to break down those hydrogen bonds. You need to separate the strands in order to read the DNA. Does that make sense to you? We good? Now, remember I told you that DNA is always read from 3' to 5'. So here's my question. Which of these strands is going to be read? That's the direction of transcription. Which of these strands is going to be read is if reading is always 3' to 5'. The bottom one, yeah. This bottom one is called template strand. And very surprisingly, the top one is called non-template strand. So what we have here is the initiation complex. This is the stage of transcription that is called initiation. Does that make sense? RNA polymerase starts to slide on the DNA molecule. And this is how in the middle of elongation, we have a picture that is something like this. So we've got this it's an official term, transcription bubble. We have RNA polymerase. Okay, moves this way. And we have RNA molecule. We have RNA molecule kind of hanging off. Can you see that? So this is elongation. That's the snapshot of elongation. What are key things? What are the key takeaway things here? First of all, how many strands of DNA are in actuality read? How many strands are transcribed? One. Yeah, only template strand is transcribed. Non-template is not. Is that clear? How many RNA strands are produced? One. Yeah, look, I'm not. If I will ask you a hard question, I'll tell you. Those questions are simple. Now, you may think, okay, if we need only template strand, why don't we just get rid of the second strand altogether, right? I mean, turns out, and still it's not clear why, but it's a law of nature, fundamental law of nature. Single-stranded DNA in nature is not transcribed. It's always double-stranded DNA. So for transcription, you need double-stranded DNA, right? Only one strand of which is transcribed into single strand of RNA. Does that make sense? Now, last bit, I'm not going to draw it, the termination. RNA polymerase runs into the so-called terminal repeats, stutters, falls off, transcription's over. Do these steps make sense? Good? Now, what's the difference between bacteria on one hand and prokaryotes in general? But I keep telling bacteria it's fine if you're not focusing on that. Bacteria on one hand and eukaryotes and viruses on another. Look at bacterial RNA. When bacterial RNA comes out, it is ready to get translated. Am I clear? So what you will see as bacterial RNA comes out, I'm going to remove the 2,5'. The ribosomes bind to it immediately, okay, and start producing protein. Okay, so that's protein. And look, this is like, imagine this. If you look at this image here, this picture, this is a snapshot, okay? So like if Kenna would be running down the hallway, and I'll take a picture of her running, I don't know where she started. I don't know where she's going to. I just have a snapshot of a person running. Does that make sense? So here, it's kind of a photograph of the whole process. But what's important? Transcription is in the process. RNA is being actively synthesized, but it is already translated, being translated. Does that make sense? So like, I'm writing a book, and you are reading it, and I still have no idea how I'm going to finish it. Like, I publish it in my blog, whatever, and you read it as I write it. Like, I develop characters, but I haven't even completed, but you already read it. Does that make sense? Same idea is here. So that's important. That can happen only in prokaryotes, because in prokaryotes, RNA is ready to be translated right off the bat, and there is no membrane that surrounds DNA. Remember, nucleoid is just DNA. Do I make sense here? So transcription and translation in prokaryotes are practically simultaneous. Now, what about viruses or eukaryotes? Here's the problem. In viruses, and in some viruses, and virtually all eukaryotes, there's a lot of what we call junk DNA. DNA that does not encode any proteins. Does it make sense? So when such DNA is transcribed into an RNA molecule, what you have is this. Your RNA molecule will contain fragments that encode protein and fragments that do not. These red ones, non-encoding, called introns. And blue ones are called exons. And I'm going to add one more. Continuing with our cookbook analogy, this is what it is. Imagine that in the recipe of a peach cobbler, you read the recipe, and then there is a fragment from Romeo and Juliet. And then you keep reading, and then there is a fragment from King Lear. Make sense? Like, it's some gibberish. So when you cook, you need to cut those fragments out. Make sense, right? Mm-hmm. So what happens is this RNA, and I deliberately don't call it mRNA, this RNA, this transcript, is going through splicing. Okay, 1, 2, 3, and 4. So those introns are removed. And what you end up with is something like that. Does that make sense? You remove meaningless fragments, leaving only meaningful ones. So then you can call it mRNA. Does that make sense to you? We're clear. So cut out that gibberish. Now, do you have to put together all 4 exons? Not necessarily. In the process that is called alternative splicing, imagine that instead of 1, 2, 3, 4, you have 1, 2, 4. Can it produce a functional protein? Yes. Will this 1, 2, 4 protein be different from the full length? Absolutely. For instance, your full length may go to a nucleus, and 1, 2, 4 may stay in the cytoplasm. Does that make sense? You understand? Like, kind of, you have peach cobbler with blueberries, but if you don't add blueberries, it's just a peach cobbler. Still, a dish. Make sense? Again, viruses and eukaryotes are capable of it. Now, remember what I said about this, the immediacy of translation. Now, look at eukaryotic cell. You make transcript, which is in the nucleus. Are you with me? You have to process it, which happens in the nucleus. Then you have to transport mRNA from the nucleus into cytosol to get mRNA translated. So you have both time constraint splicing first, and space constraint, because transcription is in the nucleus and translation is in the cytosol. Does that make sense? This is why in eukaryotes, transcription and translation are separated one from another, in time and in space. Does that make sense here? Follow me? Okay. Any questions on splicing? It's pretty straightforward. Okay. We're going to talk about translation now, and it's a rather, it's not a challenging concept. It's challenging to explain translation using the whiteboard. So this is what I'm going to be doing. We're going to say a few words about the genetic code, about tRNA, anticodons and such. Then I will explain the process of translation on the whiteboard. And then, don't worry, there's not going to be anything embarrassing. We're going to do a small reenactment. I kind of see a skepticism, but believe me, once we do it, and everybody's going to participate because we have only 5 people, once we do it, it will immediately go. It's years of practice, okay? So, translation. First of all, we need to define an idea of a genetic code. So, imagine, let me look something up really quickly. I don't even need my lectures. Okay. No. Okay, so here's what I'm going to do. I'm going to write down some absolutely from the ceiling sequence of nucleotides, okay? So the first one will be okay. AG. Let's say just U. I'm just making it up, and no. Okay, that makes sense? That's your RNA sequence. So genetic code is essentially a language of nucleotides. Now let me ask you first, how many different nucleotides we have in the RNA? You can just list them. So what are the nucleotides? What are the letters? A, U, G, C. So 4 letters. Now how many words do we need? We need words for all amino acids. We have 21 amino acids, so at least 21 words, okay? Bit more complicated than that, but it's okay. We need a word for start, and we need a word for stop. Where translation starts, where translation stops. So I'm going to put A. Okay? So we need a word for the start, we need a word for the stop, we need the word for all amino acids. So if we make words that consist of 2 letters, like UA, AG, that'll give us only 16 combinations. That's not enough. We clear? So 3-letter words give us 64 combinations, which is plenty, and actually more than we need. So some amino acids will be encoded by more than 1 word. Is that clear? So for instance, for phenylalanine, I have this UUC. Another codon will be UUU, also phenylalanine. Does that make sense? So like you can call something large or huge or big or enormous. It's all synonyms. Do I make sense? So these synonyms make genetic code redundant or degenerate. It's the same idea. Okay? Now, how does this work? So, AUG. What is AUG? It's a start code. Okay? What is UAG? It's a stop code. Does that make sense? That's where it starts, translation. It's where the translation starts. Now, AUG also encodes, I want to check myself. Yeah, it's AUG. AUG also encodes an amino acid methionine. So we have transport RNA, which has so-called cloverleaf structure. I'm not going to draw the cloverleaf, like a cross, okay. So transport RNA, which carries on its distal end, carries methionine, okay? So how this tRNA can recognize this codon? Remember complementarity. AUGC, remember that? So it turns out one of the ends of tRNA, there is so-called anti-codon, which is complementary to AUG. What will be three letters that complementary to AUG? Yes. So that's your, well, let me put it in the green. So you're absolutely right. UAC. Okay? So this is how tRNA knows. This is how proper tRNA is incorporated right here. Does that make sense? What's going to be the tRNA? What's going to be the anti-codon for the next one? For alanine. What will be the anti-codon right here? C, very good. CGA. And here it will be GUA. Okay, I'm just trying to make sure CAB. Cysteine, and here it's going to do what? That makes sense? So this is how these tRNAs know where to go. Now, please don't take it that they have a brain. No. They don't have a brain. There are no little Oompa Loompas in ribosome just pulling the right tRNAs from the solution. If anti-codon does not match a codon, tRNA cannot get in there. Does that make sense? It will not form the pairs. It will not stick into the ribosome. Are we clear? And then there is a stop codon. We'll get to the stop codon later. This whole story, does it make sense for you? Now, what you may notice is that code works. There's no overlaps. There's kind of a frame. Does that make sense? So you have 3, and then you kind of move a frame. 3, frame, 3, frame, 3. It's called a reading frame. Does it make sense? It's like you have a little window that you move over the text to select specific words. Got it? So, to kind of summarize this part, each amino acid is encoded by a codon which consists of 3 nucleotides. And one amino acid can be encoded by several different codons, but each codon uniquely encodes only one amino acid. So UUC will always encode phenylalanine. Am I clear? Okay. Let's talk about translation. It's not really easy. It's because of the amount of drawing. So what do we need first? We need mRNA. Now, how does ribosome know to bind? So in eukaryotes, it's called 5' cap structure. It's a structure on the 5' end of mRNA, right here, that serves as the ribosomal signal for binding. Okay, so ribosome recognizes 5' cap, starts binding. We'll get to ribosome in a second. In prokaryotes, similar signal is called Shine-Dalgarno sequence. Okay, so all in all, if we're going to be talking about prokaryotes mostly. So ribosome recognizes Shine-Dalgarno sequence, and the first thing that happens, a small ribosomal subunit, right here. I'm going to remove the mRNA nodes here. Small subunit binds to mRNA, and the first tRNA gets in there too. Okay. Anytime. We good so far? You following me? After all this happened, a large ribosomal subunit completes the picture. So small subunit binds to mRNA, first tRNA gets in there, and then large subunit gets in there, and we've got initiation complex ready. Good so far? Now, brief note, ribosomes in eukaryotes and in prokaryotes are different. Slightly different, like structurally different. Like, truck and a minivan, both are cars, but they differ. Okay? Which is super important, because if we can specifically inhibit bacterial ribosomes without affecting human ribosomes, then, that's a great news, right? That's fantastic because we can create drugs, and we're going to talk about it immediately. So far, we good? Okay, my least favorite part of the whole story. Bear with me, because after that, we're going to do mRNA. Small subunit, large subunit. Now it turns out in the large subunit, there are 3 sites. A site, B site, and E site. Okay? I usually kind of mnemonic. A is attachment, P is protein, E is exit. So imagine what happens here. So you've got a tRNA in the P site with a first amino acid. Does that make sense? That's your initiation. So then, remember what A stands for, attachment. Based on the match between anticodon and codon, A site kind of accepts next tRNA with a second amino acid. Does that make sense? So far, you follow me. What happens next? Amino acid 1 gets transferred to the amino acid 2. Look at this. It's kind of dynamic story, so I'm going to erase amino acid molecule. I'm going to erase that area. And now we have this. Does that make sense? Got it? Now take a look. Take a look. Turn on your imagination. Ribosome, the brown part, shifts to the right. I'm not going to redraw the ribosome. Look what happens next. After the ribosome shifts to the right, empty tRNA that doesn't carry any amino acid anymore ends up in the T site, in the E site. Does that make sense? tRNA with 2 amino acids, 1 and 2, ends up in the P site. A site is empty, and what gets in there now? What gets in the A site? What gets in the A site? What happened at the previous step? What got in the A site? What kind of RNA? It brings amino acids. So what kind of RNA? Which RNA carries around amino acids? It's tRNA, right? So here comes tRNA with amino acid 3. Now look what happens next. We're not going to redraw it massively, but this dipeptide gets transferred here. Does that make sense? I put it as exit because this empty tRNA just gets out of here. Okay? Now, here's the thing. First of all, well, not first of all. So this goes on and on and on and on, until it runs in a stop codon, and then the release factor liberates polypeptide. Now, in order to show this to you, I have a question. All 5 of them will participate. Seriously. I have a question. Serious question. Okay, so this is what we try to reenact. These 2 chairs are A site and B site. I don't have E, it's there somewhere. Okay? All these ladies are tRNA. So these are, well, except for Angelina, but we'll get to this. So these 4 ladies are tRNAs, and the markers that they hold are amino acids. So, Nishka, get in the P site. This is our initiation complex. You see? That's the first amino acid in the P site. Now, second tRNA gets in the A site. Now the transfer happens like this. Okay? This tRNA is empty. This tRNA now carries dipeptide. Ribosome shifts. I'm not going to keep the chairs from under you. So we're going to remember that shifts. So this tRNA, empty tRNA, leaves. Thank you. And tRNA with a growing polypeptide gets in the P site. The next tRNA comes. Excellent transfer. The ribosome shifts. It shifts. Okay. Next tRNA comes in. Transfer. The ribosome shifts. And now come the release factor. Look at the release factor. So release factor comes in. There is no transfer. Release factor has this red flag sort of. And what this release factor does, because there's a stop codon there, it liberates polypeptide and the synthesis is done. Does that make sense? Better? Awesome. Good. Thank you so much. Good. Okay. So we kind of covered this to mention a few more things. Thing number one, this whole process requires energy. So ATP, a lot of ATP. If my memory serves me well, 2 molecules of ATP per amino acid. Okay. Second, I don't know if you remember your previous bio classes. You may have been told that there are 20 amino acids. There's 21. If somebody is willing to bet $100 against me, go for it because you're going to lose. 21 amino acids. In addition to classic 20, there's an amino acid that is called selenocysteine. It's like cysteine, but instead of sulfur, there's selenium. It's not a very frequent amino acid, but it's crucially important for various enzymes that deal with oxidative stress. It is thought to be involved in regulation of aging in cells and blah, blah, blah. What's interesting about selenocysteine is that it is encoded if, let me switch and check something. I think it is encoded by UGA stop codon, although I would not take my words to the bank. The point is, amino acid is encoded by a stop codon. How can it be? Well, let me show you. Imagine that you have an mRNA, okay? And somewhere here, you have a stop codon. If ribosome moves down mRNA and sees the stop codon, what it should do? It's not a hard question. If it sees a stop codon, what it should do? Stop. But if there is a special sequence behind stop codon that is called selenocysteine insertion sequence, ribosome recognizes that sequence, and instead of stopping, introduces selenocysteine and keeps going. Does that make sense? My favorite analogy, you go down the street, there is a sign, road closed. What do? You turn around. Or whatever, you turn around, you find a detour. So that's your stop codon. You go down the street, there is a sign, road closed, but by that sign is the police car, and the officer tells you, keep moving, you find. So that police car is selenocysteine insertion sequence, that police officer. It tells ribosome, instead of stopping, put selenocysteine and keep going. Does it make sense to you? Okay, got it? We have last very small bit. Comparison of gene expression between you and prokaryotes. Okay, so ribosomes are different. This 70S, 80S bullcrap, you don't need to memorize the numbers. It's not important. I want you to understand ribosomes are different. Does that make sense? In prokaryotes, transcription and translation are approximately simultaneous. Remember that picture with ribosomes jumping on RNA that is yet being synthesized that I had? Remember? In eukaryotes, transcription and translation are separated in time and space. So basically, what I'm talking about here, you have nucleus, you have splicing, there's no splicing in prokaryotes. That makes sense? There's no RNA modification in prokaryotes, a lot of RNA modification in eukaryotes. And finally, monocystronic transcripts What does that mean? Look here. So this is DNA molecule with 3 genes. Gene 1, gene 2, gene 3. And the DNA molecule here. Gene 1, gene 2, gene 3. We're good so far? So what RNA is going to look like in eukaryotes? Three separate transcripts. Each of them contains only 1 gene. Monocystronic, monocystronic, monocystronic. Does that make sense? What the transcript is going to may look like in prokaryotes. So mRNA, single mRNA, contains 3 genes, 1, 2, and 3. Does that make sense to you? Questions? Stella.